This method is helpful to detect and co-localize multiple biomarkers in a single cryosection, including markers that are not normally detectable in paraffin sections. Using this experimental staining method, the staining time is significantly reduced, and the method can be performed in any laboratory. Demonstrating this method will be Dinesh Jaishankar. He is a skilled postdoc from the lab and also the Immunotherapy Assessment Core Facilities Manager.
When selecting antibodies for the analysis, consider the excitation and emission filter sets available on the semi-automated imaging system, and, after testing various combinations of fluorophore-conjugated primary antibodies, prepare fluorophores to be used that have minimal spectral overlap, as suggested in the table. For multiplex staining, prepare a cocktail of antibodies with compatible fluorophores at predetermined optimal dilutions, and add the cocktail to the tissue sections of interest. For single marker staining, add the primary conjugated antibody only to the slide. For all stainings, include a control unstained slide that undergoes the same staining procedure without the addition of a primary conjugated antibody. After a 1-hour incubation at room temperature protected from light, wash the slides 2 times with PBS for 5 minutes per wash. After the second wash, counterstain the multiplex stained slides with DAPI for 7 minutes at room temperature protected from light, followed by 2 5-minute washes in PBS. To create a spectral library, set the lamp power of the multispectral